That doesn't necessarily preclude the possibility that an early population of Homo habilis could have given rise to Homo erectus, but it does imply one of two things. So it either implies that this divergence happened much earlier than we actually have record of in the fossil record, and we call that a ghost lineage. So that means it must go much further back in time than we have sampled. Or it means that there was a very rapid evolutionary shift to go from something like Homo habilis, let's say around 2.5 million years ago, with that relatively primitive body plan, to this very modern looking Homo erectus body plan just 500,000 years later.
Dr. CARRIE MONGLE, thanks so much for coming back onto Evolution Soup all the way from your office in New York, USA. You are an assistant professor in the Department of Anthropology and Turkana Basin Institute at Stony Brook University, and your research focuses on the discovery and description of new hominin fossils from the Turkana Basin in Kenya. In our last interview, you told us about the discovery of new Paranthropus boisei fossils from the Koobi Fora site in Kenya, including hands and feet fossils. Today, we return to Koobi Fora to examine an incredible find, the most complete skeleton of the ancient hominin, Homo habilis, found so far. Welcome back, CARRIE. Well, you just recently returned from Turkana, and of course, the new paper on habilis has just been published. What with that, the Paranthropus discovery, and battling the snow in New York, it's it's been all go lately for you, hasn't it?
Yeah, thank you so much for having me back. We're really excited to have this paper out. I certainly cannot take all the credit. So, just like the last paper, this research was really the culmination of a team effort, and in this case, led by Dr. Fred Grein, who's also at Stony Brook. And the nice thing about doing a skeleton like this is each member of the team can bring a different expertise to the project, and we're each responsible for our own little parts of the analysis. And then, of course, none of this research is possible without ongoing years and years of work by me, even Louise Leakey, directing the Koobi Fora research project.
The Homo habilis fossil is the best, most complete habilis postcranial skeleton that we have to date. CARRIE, can you give us an overview of habilis, its discovery, the early fossils, as well as initial conclusions about the species?
Yeah, I'd be happy to. So, the species Homo habilis was first named in 1964 by Louis Leakey, and this was based on a fossil called OH7, which stands for Olduvai Hominid 7, of course, from Olduvai Gorge. This fossil included a jaw, some hand bones, and some skull fragments. And what was especially striking at the time was that you see this combination of a slightly larger brain than the Australopithecines that we'd been seeing, paired with smaller teeth and a hand that appeared capable of making stone tools. And so at the time, that discovery was really transformative. Prior to OH7, Paranthropus had been discovered in those same Olduvai deposits and had been the assumed toolmaker. This new fossil, just found a few years later with its smaller teeth and bigger brain, effectively dethroned Paranthropus for a while as the toolmaker at Olduvai. And what's really interesting, I think, when Leakey and his colleagues were naming the species Homo habilis, which literally translates to handyman, they were doing more than just describing the anatomy. They were tying toolmaking directly to what it meant to be a member of our genus, the genus Homo. And of course, now we know the story's a bit more complicated and that there are tool-making hominins around well before early Homo. But I think in this early definition of kind of what does it mean to be the earliest member of the genus Homo, we were already thinking about kind of what qualifies a hominin to be considered more closely aligned with us, Homo sapiens, than the earlier australopithecines that had come before. So in the next few decades, additional fossils have expanded the sample, but as always, it also complicated the story. So one particularly important specimen for this was one called OH62. So again, Olduvai hominid number 62, and that was described in 1987. That fossil included some fragmentary limb bones, and what was really surprising about it is that it showed that Homo habilis seemingly had these Australopith-like limb proportions, so relatively long arms and relatively shorter limbs compared to what we think of as modern human proportions that we all have today. So not exactly that kind of tall, long-legged, human-like body that we see really in Homo erectus. And so that, again, raises this question of how human does this early Homo really look, and what does it mean to be considered part of the genus Homo? So I think, with this body proportion in mind, this really leads us to the question of what does it mean to be a member of the genus Homo? And this has sparked a huge amount of debate over the years. So there are some taxonomists who would say all hominins count as genus Homo, and others who would say it's really just Homo sapiens and maybe Neanderthals. So I wanted to go through a bit of a definition of how we go about drawing that line. From just a purely cladistic standpoint, a genus is defined as a last common ancestor and all of its descendants. That could be a single species or that could be a group. Um, so again, phylogenetically speaking, genus Homo can include Homo habilis and Homo rudolfensis and Homo erectus because those are all more closely related to one another than they are to other hominins. And I personally fall into that category of, I think as long as it fits Linnaean taxonomy, then by all means it's fine. But there are other kind of theoretical biologists who have argued that a genus should also represent an adaptive plateau or be kind of diagnosably distinct from other genera. And so in that respect, that's where the real theoretical debate comes up of what does it mean to be adaptively distinct and more similar to Homo sapiens than to earlier hominins. So is it our bigger brains? In which case Homo habilis kind of barely makes the cut, but it's still there. Is it our smaller teeth? Um, you know, in that situation, people would argue that Homo habilis still has relatively large dentition compared to later Homo. Um, is it our body plans? Is it, that we've got this very different body plan than we saw in earlier hominins? And that's really the case for some people saying that Homo habilis should be kind of relegated down to Australopith status. Or is it something else? Originally, when Homo habilis was defined, it was considered that tool making was the thing that made a hominin a member of the genus Homo, or at least that was part of the package. And so it really is a moving target. And I think it's more of a philosophical question than a scientific one of what does it mean to be a member of our own genus. And again, I personally feel like Homo habilis makes the cut, but I know there are others who might disagree with me on that now.
The first fossils of this skeleton were found back in 2012 in Kenya as part of the Koobi Fora research project, directed by Meave and Louise Leakey. Bit by bit and year by year, more bones were found. So, what exactly was unearthed, and why are these bones so important?
So in 2012, Arbolo, who's one of the members of the Koobi Fora research project, recovered the first tooth of this specimen during some routine surveys on the eastern side of Lake Turkana, a bit north of Koobi Fora from where the hand was found. So we're on the eastern side of the lake, but again, further north than the Koobi Fora research area. Subsequent excavation in that particular location revealed the rest of a complete lower dentition, so all of the teeth that would go in the lower jaw. And then scattered down kind of a nearby slope, they started finding just hundreds of small skeletal fragments. And Dr. Deming Yang, who is now a professor, but at the time was an intern working with Meave Leakey, started putting all of these little fragments together like a 3D skeleton puzzle. And so as he starts piecing this together, it becomes clear that there's a hominin skeleton, or at least part of a hominin skeleton. So then the question for the team became, does that skeleton go with those teeth that were found just a few meters away? Occam's razor would suggest that they do, since hominin fossils are so rare and these were within a few meters of each other. They seem to be the same developmental age. They're both adults. But the team wanted to be absolutely sure. And so one thing Fred Grein noticed when he and Meave Leakey took the fossils to micro CT facility in South Africa was this really bright inclusion on the scans of both the teeth and the bones. And that suggested that there was some kind of very dense mineral that had infiltrated during fossilization. So Fred started to wonder, maybe this could be key to associating the remains to a single individual. Presumably, if all of these elements belong to a single individual, then they would have been buried and fossilized in the same depositional event, meaning they're exposed to the same mineralization process. So if that's the case, then you would expect their geochemical signatures to be the same. So this became a project unto itself. A team of geochemists were enlisted, and this was led by Dr. Ted Present at Caltech. And he was able to publish in 2024 this association based on the geochemistry of both the teeth and the bones that they most probably do go with a single individual. So prior to the publication of the full skeleton, we actually, in 2019, had already published the dentition because we could, just from the dentition, association aside, tell what species this was. So luckily with hominin teeth, they tend to be quite diagnostic. And you can look at the kind of the size of the teeth, especially the relative sizes of the teeth. So how big are the molars relative to the premolars? How big is the first molar relative to the third molar? And also kind of the configuration of the cusps and what cusps are present to tell you something about what species this is. In our case, this dentition compared really favorably with the type specimen for Homo habilis. So we already had that established. We already knew what species it was. Then with the geochemistry, we had to figure out if the skeleton went with the Homo habilis dentition. And of course, like I said, in 2024, Ted Present and other geochemists were able to make that association, which is why now we've been able to finally publish this as the most complete skeleton of Homo habilis, not just of an early Homo skeleton.
So, what does the anatomy of this habilis skeleton tell us?
If we think about modern humans today, we have relatively long legs and relatively short arms. And you see this in the fossil record with other species like Homo erectus. When we look at something like Homo habilis with previous fossils like OH62, we had these hints that that wasn't quite the case in the earliest members of the genus Homo. So it seemed to suggest that, at least from OH62, which again was quite fragmentary, that Homo habilis would have had body proportions that were more similar to Australopithecines, so relatively longer arms and relatively shorter legs. This is a much more complete upper limb skeleton that really allows us to confirm that previous hypothesis. So our analyses show that Homo habilis did in fact have relatively long forearms. And when we estimate stature from the preserved pelvis, or the small fragments of it that we have, this individual would have been considerably shorter than Homo erectus. So, like I said, we thought this before based on OH62, but I think what's really exciting about this skeleton is that it preserves so much more of the upper limb and really allows us to confirm those earlier suspicions with much stronger evidence. So taken together, I think, this suggests that early members of our genus had a body plan that looked much more like early hominins, like someone like Lucy, and much less like us. So it's not until Homo erectus that we start to see this very human-like body plan.
And what about evolutionary implications? Are we seeing Homo habilis in a totally different light?
So conventional interpretations of the hominin tree have always placed Homo habilis as more or less directly ancestral to Homo erectus and later members of the genus Homo. We now know that they overlapped in time, so Homo habilis and Homo erectus would have overlapped in time because Homo erectus was around by at least 2 million years ago. And this suggests that the two species coexisted on the landscape. That doesn't necessarily preclude the possibility that an early population of Homo habilis could have given rise to Homo erectus, but it does imply one of two things. So it either implies that this divergence happened much earlier than we actually have record of in the fossil record, and we call that a ghost lineage. So that means it must go much further back in time than we have sampled. Or it means that there was a very rapid evolutionary shift to go from something like Homo habilis, let's say around 2.5 million years ago, with that relatively primitive body plan, to this very modern looking Homo erectus body plan just 500,000 years later. And then thinking about what was Homo habilis doing and how were they partitioning the landscape, there's always going to be a leap to think, oh, they had very different body proportions, so they must have been, maybe one was more arboreal than the other. Maybe they were niche partitioning in a particular way. But I think, that brings up the question of, did they have these traits because they were selected for? In that case, a functional explanation of why they have such strong upper limbs. Or do they retain these traits because they've not yet been selected against? And I think that's likely the case in this situation where you have this australopith like body plan still in Homo habilis because it's not yet been selected against.
Well, the first fragments of this hapless individual were found 14 years ago and was announced to the world just recently in January 2026. The question that people often ask is, why does it take so long?
Of course, that's completely understandable. So as a bit of a backstory on this particular fossil, as I said earlier, we published the dentition in 2019 and then spent the next few years with a team of geochemists trying to sort out the association, whether we could publish the postcranium as associated with Homo habilis dentition or if it was just going to be kind of taxonomically unidentified. But I think, that doesn't explain 14 years. So I wanted to just go through a little bit of the process of kind of recovery to publication for one of these fossils. So, for a fossil discovery like this, the excavation alone can take years. People see the picture of this nice, beautiful reconstruction, but what they don't see is that each of those tiny, tiny fragments was carefully excavated out of the ground and also mixed in with a bunch of other kind of faunal, crocodile teeth, bovid bones, whatever. So it takes a huge amount of time to just move that amount of dirt and to do it carefully and in a really documented way. Once everything is out of the ground, fossils can often be very fragile. And so the preparation process takes again years, and in this case we have a team of preparators, but when the fossils are this fragile and this fragmentary and this important, there's really only one or two people that we can have work on it, and so that takes the time that it takes for him to, with a Dremel, like just very carefully and slowly go through that. And the saying slowly, slowly reminds me because you always hear them in Swahili. It's pole pole, slowly, slowly. So once that's done, our next step is going to be to CT scan the material. But for a micro or a nano CT, and certainly for a synchrotron scan, we can't do that in Kenya. And so we have to obtain export permits. And you can imagine these fossils are part of Kenya's national heritage, and exporting fossils out of the country is not taken lightly. So that process itself can take months to a year, just making sure that everything is properly in place for that travel and making sure that it's going to be in the hands of the Kenya National Museum as soon as it gets back. So that process takes time. Then you have to coordinate this international team of experts that are going to come together and study each piece of the skeleton and conduct all of those analyses. And it's not just an analysis of the fossil itself, it's also a matter of gathering the comparative data from other fossils and as well as from living primates. So that takes a bit of time. And then in the case of this one, I wanted to also note that we were delayed a bit because of the tragic and untimely death of Professor Bill Jungers, who was originally leading this description. So I think after that, it took the team a bit of time to regroup and try to move this forward in a way that really honored his legacy.
Well, it's been great having you back on the show, Carrie, and hopefully we'll have you back again on Evolution Soup in the near future. So, are there any projects that you're working on that you can tell us about?
You mentioned at the outset that I just got back from Turkana, so I can report there are new hominin fossils coming out of the ground as we speak. So one of my immediate plans is to get back to Turkana with Louise as soon as I can, so we can try to figure out what those are. But on the Stony Brook side of things, I'm still going to be here teaching. If students want to go out into the field with me, they can always sign up for the Turkana Basin Institute Field School program. I know there's going to be a group going out this summer as well as next spring. So plenty of opportunities for other people to get involved. As I said, there are new hominin fossils coming out, and it's a really exciting one. We're not entirely sure what it is yet. Um, and then lots of CT data processing. So, as I said, these things take years, and a lot of my time is spent behind the computer looking at all these scans and going kind of slice by slice and seeing what we can figure out about these specimens. So that's what's keeping me busy these days.
Dr. Carrie Mongle, thank you very much indeed.
Thanks for having me.